All right, so are you ready to dive deep into some cutting-edge DNA research? Because we're taking a look at this fascinating article from the University of Würzburg.
Oh, yeah.
It's called Scientists Just Discovered an RNA That Repairs DNA Damage. And it's a game changer.
Wow.
Published just this month. And trust me, the title is not an exaggeration.
I bet.
This article, it's all about this long non-coding RNA called NEAT1.
Okay.
And how it's changing everything we thought we knew about how cells repair their own DNA.
It's really remarkable. For years, scientists, we were so focused on proteins when it came to DNA repair. But now it's becoming really clear that these RNA molecules like this NEAT1, they play a way more active role than we ever thought.
So DNA, it's under attack all the time, right?
Yeah, definitely.
Every time our cells divide, there's always that chance for errors to sneak into the genetic code.
Think of it like, making a photocopy of a photocopy.
Right.
Eventually, the image, it's going to get all distorted and blurry.
Right. It's not going to look as good.
Exactly. And it's not just cell division, right?
Yeah.
There's also external things like UV radiation from the sun.
Oh, sure.
Certain chemicals. Even things like smoking. All that can damage our DNA.
Well, okay, so our cells have some kind of defense system then.
Oh, absolutely, for sure.
To fight back.
Yeah, cells have evolved these really sophisticated mechanisms to detect and repair DNA damage. It's called the DNA damage response, or DDR.
DDR, got it.
Yeah, so it's constantly monitoring the genome, looking for these errors.
Okay.
And when it finds them, it triggers this whole cascade of events to fix the problem.
So it's like a repair crew?
Yeah. It's like the cell's internal repair crew, always on patrol.
Always on patrol.
Ready to jump into action.
That's cool.
Yeah. And, the type of damage that this research focuses on is really serious stuff. We're talking double-strand breaks.
Okay, double-strand breaks.
Yeah, these are complete breaks in the DNA molecule, almost like snapping a rope in two. Yeah. And they can be caused by things like radiation or certain chemicals.
So that sounds really bad.
Yeah, so double-strand breaks, they can lead to, big genomic rearrangements, deletions, or even chromosomes fusing together.
Oh, wow.
Yeah, these changes can really mess with how genes work, leading to cell death, or even worse, uncontrolled cell growth, which is, cancer.
That is bad.
Yeah, so the cell really needs a strong system to deal with these breaks.
Right.
And that's where NEAT1 comes in.
Okay, so back to NEAT1.
Right, so what these researchers at the Julius Maximilians University at Würzburg found.
Wow, what a name.
I know, right? They found that when these double-strand breaks happen, the cell makes a lot more NEAT1. And not only that, they also found that NEAT1 itself, it undergoes a transformation.
Oh.
Yeah, it gets modified with these chemical tags. They're called N6-methyladenosine.
N6-methyladenosine.
Yeah, that's a mouthful.
So what are those?
So think of it this way.
Okay.
N6-methyladenosine, or we can just call it M6A.
M6A. Much easier.
Yeah, much easier. So it's like attaching a little sticky note to the NEAT1 molecule.
Okay.
So it doesn't change the RNA sequence itself, but it changes how it folds and how it interacts with other molecules.
Interesting.
Yeah, and this is actually a whole field of study called epitranscriptomics. It's how these little modifications to RNA can affect what it does.
Okay, so by adding those tags to NEAT1, the cell can control how it reacts to DNA damage.
Exactly. And the research showed that when NEAT1 gets heavily methylated because of those double-strand breaks, it changes shape.
Oh, wow.
Imagine like a long chain that suddenly folds in on itself.
So it's not just sitting there.
Nope, it's actively responding to the damage.
That's really cool.
So does it go in and fix the broken DNA directly?
Yeah, does it?
Well, that's where things get even more interesting. NEAT1, it doesn't actually repair the DNA itself.
Oh, okay.
Instead, it acts as a platform, a kind of meeting point for other molecules involved in the repair process.
It's more like a coordinator.
Exactly, like a construction site foreman,
Yeah, makes sense.
making sure everything's in the right place to rebuild the damaged structure.
Wow, so this is a whole new way of looking at how these long non-coding RNAs work.
It really is. It changes everything.
That's amazing. So tell me, what does this mean for treating diseases, especially cancer?
It really changes how we think about these RNA molecules. Instead of just being these passive carriers of genetic information, they're like active players in all these important processes in the cell.
That's pretty incredible. And this has big implications for how we treat diseases, especially cancer, right?
Absolutely. One of the things that makes this discovery so exciting is that NEAT1, It's often found in higher amounts in tumor cells.
So if NEAT1 is helping cancer cells repair their DNA, wouldn't that make them stronger and harder to kill?
Yeah, that's one way to look at it. Cancer cells, they divide so rapidly and accumulate more DNA damage, so they might be hijacking NEAT1 to help them survive and grow.
But couldn't we use that to our advantage?
Exactly. That's the really exciting part. It gives us a new target for therapies.
So how could we target NEAT1 to fight cancer?
Well, there are a couple of approaches scientists are looking into.
Okay.
One strategy would be to develop drugs that block NEAT1's activity specifically in those cancer cells.
So kind of disarm them.
Yeah, exactly.
Yeah.
If we can stop NEAT1 from forming that DNA repair complex, it could make the cancer cells more vulnerable to treatments like chemotherapy or radiation.
So we'd be taking away the repair toolkit.
That's the idea, but of course it's not quite that simple. One of the biggest challenges is figuring out how to just target NEAT1 in the cancer cells without messing with its normal function in healthy cells.
Right. We don't want to weaken the DNA repair system in healthy cells.
Exactly. They could do more harm than good.
So are there other options?
Well, another interesting possibility is to manipulate that methylation process we talked about. Remember those M6A tags that attach to NEAT1?
Yeah, the sticky notes.
Right, the sticky notes. So researchers are trying to figure out ways to either increase or decrease that level of methylation.
So we could fine-tune its activity.
Exactly. For example, if we could find a way to increase methylation on NEAT1, but just in the cancer cells, it might actually overload their DNA repair system.
Oh, wow. So they'd be too busy trying to fix everything.
Exactly, and that could slow their growth or even make them die.
That's really cool. It's like using their own system against them.
Right. But of course, that kind of manipulation is really tricky. We have to make sure we're only affecting the cancer cells and not harming the healthy ones.
So how do we do that?
Well, that's the big question, isn't it? Scientists are working on understanding the differences in how NEAT1 works in healthy versus cancerous cells and then designing therapies that exploit those differences.
It sounds like there's a lot more work to do.
Absolutely. But the progress that's being made is amazing. And this discovery about NEAT1 and DNA repair, it's really just the beginning.
Yeah.
It's opening up a whole new world of possibilities for understanding how our cells stay healthy and how we can intervene when things go wrong.
So this research has implications beyond just cancer.
Oh, definitely. Think about it. DNA damage happens in all cells, not just cancer cells.
So this could be helpful for other diseases, too.
Exactly. This research could help us understand aging, genetic disorders, even things like neurodegenerative diseases.
Wow. So NEAT1 could be a target for all sorts of different therapies.
Absolutely. But what's really fascinating is that this also shows us just how connected RNA and DNA are in the cell.
Tell me more.
Well, for a long time, we thought DNA was the star of the show.
Right, the blueprint of life.
Yeah, and RNA was just the messenger, carrying information from DNA to the ribosomes to make proteins.
Yeah.
But now we're starting to see that RNA is much more dynamic and important than we realized.
Like it's been hiding in plain sight.
Exactly.
Yeah.
And the fact that these modifications to RNA, like those M6A tags on NEAT1, can actually change how it behaves and interacts with other molecules. It just adds this whole new level of complexity to gene regulation.
It's like RNA is controlling everything.
That's a great way to put it. And this discovery about NEAT1 is just one example of how our understanding of RNA is changing how we think about biology and medicine.
That is incredible. It's like a whole new world is opening up.
It really is.
Yeah.
And as we learn more, it raises all sorts of interesting questions about life itself.
Like what?
Well, think about this. If RNA is so crucial for things like DNA repair and gene regulation, what does that tell us about how life began? Some scientists think that RNA might have actually come before DNA.
Wait, so you're saying life might have started with RNA, not DNA?
It's a pretty radical idea,
yeah.
right? It's called the RNA world hypothesis, and it suggests that early life forms used RNA for everything.
Wow, so RNA was doing it all.
Exactly. And even though DNA eventually took over as the main way to store genetic information, RNA kept its important roles in all these different cellular processes, like we're seeing now with DNA repair.
So RNA has been a multitasker from the very beginning.
Absolutely. And who knows what other secrets RNA is hiding.
I know, right? But let's talk about those RNA-based therapies for a second.
Yeah. We talked about how this research on NEAT1 could lead to new treatments, but what are some of the challenges in developing these therapies?
Well, one of the biggest challenges is figuring out how to deliver those RNA molecules to the right cells in the body. RNA is fragile. It can be broken down easily by enzymes in the bloodstream.
So we can't just inject it?
Not quite. Scientists are developing all these different delivery systems to protect the RNA and make sure it gets where it needs to go. Things like packaging the RNA in nanoparticles or using modified versions of RNA that are tougher.
So it's like a microscopic delivery truck?
Exactly. And then once the RNA gets to the right cell, we have to make sure it actually gets inside and reaches the right machinery to do its job.
That sounds complicated.
It is. But even with all these challenges, the field of RNA therapeutics is moving really fast. There are already some RNA-based drugs being used, and lots more are in development.
That's good news. But are there any risks with this new technology? Well, of course, with any new technology, we have to be careful. There's always the chance of what we call off-target effects, where the RNA might accidentally interact with other molecules and cause problems.
So we need to be careful and do lots of research.
Absolutely.
Yeah.
We need to ensure any new treatment is safe and effective, especially something as powerful as RNA therapy.
So this has been an amazing deep dive into the world of NEAT1 DNA repair and the amazing world of RNA.
Yeah, it has.
What's the one thing you want our listeners to take away from all this?
Yeah, I think the biggest takeaway is that we're only just starting to understand RNA,
Yeah.
This molecule that we thought was just a messenger, it's actually a key player in so many cellular functions.
Like keeping our DNA healthy.
Exactly.
And this discovery could completely change how we treat diseases.
Absolutely. It opens up so many possibilities for new therapies that target RNA.
It's really exciting.
It is. It's a really exciting time for biology, and I'm sure we'll learn even more about the amazing world of RNA in the years to come.
This has been an incredible deep dive into the world of NEAT1 DNA repair and the expanding role of RNA in our cells. What's the key takeaway you'd like listeners to remember from our conversation?
I think the most important message is that our understanding of RNA is completely changing. This molecule that we used to think of as just a messenger is actually a central player in so many cellular functions, including, keeping our DNA healthy.
And this discovery could totally change how we approach treating diseases, opening up all sorts of exciting new possibilities for therapies that target RNA.
It really is a fascinating time for research, and I'm sure we'll keep uncovering amazing new insights into the world of RNA in the years to come.
Well, thank you so much for taking us on this journey with you. It's been eye-opening, and I have a whole new appreciation for how complex and amazing life really is. And to all our listeners out there, we encourage you to keep exploring this topic and stay curious about the world around you.
Absolutely. There's always something new to discover.
And that's it for this deep dive. We'll be back soon with another exciting look at the world of science and discovery. Until then, keep on diving deep.